Good morning. Thank you for joining us today. I want to first thank ARM and all the sponsors actually of the meeting for allowing us to be able to present our various technologies. It's always great to be able to mingle with peers and to exchange ideas. My name is Raphael Ognar. I'm a CEO and co-founder of NKILT Therapeutics, and I'm going to give an update on where we are with our novel technology, targeting a unique target called HLA-G with our chimeric ILT receptor technology. The way we started the story of NKILT was based on looking at all the details of what happened in cell therapy in the past decade, particularly. Since the first CAR T came up into patients, we made significant progress, but we also hit severe road bumps, particularly when we tried to expand beyond T cells, when we tried to expand beyond B-cell malignancies, diseases, and when we tried to reach solid tumor. And our belief, it's because we need to be more fit for purpose on how we build and engineer our immune cells. The second key component of our thinking process was autologous therapies are great today with whatever we can deliver through this technology, but we're not going to be able to scale up this to very large populations in the near future, and we're also not going to be able to sustain such costs that we have today. We believe that going through an allogeneic technology to develop true off-the-shelf approaches will help in the meantime while we are trying to figure out how to go into the holy grail of in vivo cell therapy. So based on this, NKILT Therapeutics created a new approach using natural receptor engagers by building a chimeric ILT receptor that is specific to the target HLA-G, which is a target that you will see is very exciting and interesting because it's not only expressed on the tumor, but also in the tumor microenvironment. And we think our KIR-NK engineered cells will be able to bring new opportunities for patients in the treatment of cancer. So we are now set to go for our IND. We have enough initial data that allows us to be confident that we have a path to IND. And the team that we assembled with my co-founders, Henry Bell and Simon Wayne Hobson, we have also two great board members that have deep expertise in cell and gene therapy and that are really helping us steering the company. And in addition, our scientific advisory board, chaired by Dr. Sargil, gives us a very strong opportunity to really bring the top thoughts into the process and support our way of developing the new therapy that we want to bring together. And it is really feeling very grateful for us to have people of that caliber that are understanding and supporting us in a journey that is, of course, full of bumps, but will be also very exciting. So 2 words on HLA-G as a recap for those who haven't really seen us before, but HLA-G is known to be the pregnancy molecule. It is actually a pretty efficient immunosuppressor that is used by the fetus to inhibit and neutralize all the immune system of the mother. So I guess we are all here in this room the proof that it works pretty well, like other 8 billion people in the world. Well, what happened is that cancer cells have figured out that nice mechanism, and over 50% of cancer cells across multiple tumor types are expressing HLA-G using that same mechanism that is leveraging the ILT2 and ILT4 receptor that are potent inhibitory receptor triggering an immunosuppressive effect that shuts down all arm of the immune system. The other interesting aspect of HLA-G is that besides pregnancy, where it is heavily expressed by the placenta cells, it is almost not expressed or very little expressed in normal tissues, which make it a good candidate to limit the risk of on-target off-tumor effects. As I mentioned, HLA-G is expressed in over half of cancer cells. On the left-hand side, you have a table that is highlighting some of the expression levels across multiple cancer, which makes it a really strong ground for us to develop new approaches and try to bring more options for multiple cancer patients. The other characteristic I highlighted briefly initially is that HLA-G is also expressed in the tumor microenvironment through DC10 cells or even some Treg cells, which kind of bring this, if I were to illustrate a little bit, this kind of immunosuppressive blanket reinforced into the tumor microenvironment and making in many aspects the tumor microenvironment a potent immunosuppressive environment. So we believe that with our approach, our KIR NK cell will not only be able to, of course, identify the cancer cells and not be fooled by the expression of HLA-G and use that against the cancer cells to trigger cancer cell killing, but we also think that they will be able to eventually modify some components of the tumor microenvironment to eventually try to reconvert it into a pro-inflammatory environment, which will allow to not only have our cells killing cancer cells, but eventually associating it with other immunotherapies. So another key and interesting factor that made some people thinking HLA-G was a difficult target is that it is actually expressed in seven different isoforms. So if you go with an antibody and you target HLA-G1, as soon as the cancer cells are going to express other isoforms of HLA-G, your antibody is going to find some limitation because, as you can see in the cartoon illustration, the structures can be significantly different between the isoforms. And that will make your antibody falling short at some point, which will also be one of the bases of what we call tumor escape mechanism that HLA-G is deeply involved in. So with that in mind, when we were using a cell engager, natural cell engager, using the protein ligand instead of using an antibody, we actually can go around this potential issue, and we had actually preliminary data in the lab that were showing very interesting results in that sense. So this is a cartoon that shows a little bit in a very simplistic way how the engineering is happening. On the left side, you have HLA-G in its dimeric form, which is actually the active form of HLA-G. And you can see that through the ILT2 or ILT4 receptors that are here in blue, you have a immune cell inactivation through those potent inhibitory receptors, as I mentioned to you. On the right side, this is an illustration of how our construct could be looking like, where we have removed some of the domains of ILT2 and ILT4 and replaced this with a stalk, and leading to our activation domains that of which I will detail a little bit more. We have many options available. Will literally revert this inhibitory effect into a NK cell activation effect. So on the left side is just another illustration with a different, ILT2, ILT4, showing that you can have like really different construct. But this is some of our preliminary data where we were highlighting how our KIR NK cells were able to target and kill HLA-G positive AML cell lines. Here we have two types of cell lines, Kazumi1 or MOM13, in which you had expression of HLA-G. And you can see that our, not only you have, on the left side, what we call the MOC, which is normal natural killer cells that are known to be good at killing leukemia cells. So we wanted to use this as a good positive benchmark. On the right side, you have the tumor only. And you can see here that several of our clones that we initially had were really strong at eradicating the cancer cells, the leukemia cells expressing HLA-G. And the interest is we did that in 6 different leukemia cell lines, and we always obtained similar outcome. Just for the records, this is at the ET ratio of 1 to 10. We actually today have pushed the data to a 1 to 20, and even in some cases 1 to 30. And our cells are truly serial killers, able to eradicate those leukemia cells. This is another piece of data that is highlighting a little bit more the differentiation versus our KIR construct versus a standard antibody-based construct or CAR construct. You have on the left side expression of cell lines with HLA-G, and you have some of our KIR construct and a standard SCAV HLA-G CAR, which you can see is showing some significant impact also in cancer cell killing, but in some situations, particularly when it was on the longer term, you started to see the CAR having some difficulties, the CAR NK, to continue to kill cancer cells. On the right side, we pressure test a little bit more, and in this case, on the right side, it's expression of interferon gamma as a marker of cancer cell killing. And you can see here that on a THP1 HLA-G1 expressed cells, you would have our KIR, in that case KIR5 clone, and the HLA-G CAR really strong at targeting and releasing interferon gamma that will be the proof of cancer cell killing. But if you look on the right, the same experiment within THP1 HLA-G2 expressed cells, our KIR remained strong outcome while the CAR was starting to also fade in terms of being able to kill the cancer cells. This is very early data, of course, but it's a very good trend for us to pursue once we're going to be reaching eventually also the solid tumor area. A little word on our safety, preliminary safety. So what is really important when you bring a novel technology is to ensure that you're not hitting on things that you were not expected to hit. And so what we are doing is a broad primary screening through human cells that you have some here that are pretty critical, like cardiomyocytes or hepatic sinusoidal endothelial cells, which, you know, if you start to destroy those, you're going to be in serious trouble with your therapy. And we tested our engineered cells across multiple of those normal human cells. And you can see in an illustration of 6 of those here that we have not seen any unexpected toxicity coming out yet from this first generation of engineered cells. And we are continuing this screening with over 20 different normal tissues. And of course, we will go into in vivo assessment in the coming months. But this was a very strong positive signal for us to be sure that our cells are not affecting normal tissue. Last point on data, very important point. We have developed a proprietary co-activation domain. We actually have multiple of them. I will encourage you to go see the publication from our CTO from previous days where he was showing how activating using MIDI88 signaling will allow to activate NK cells beyond 2 weeks in a settings in vivo where they will be able to be active for up to 6 weeks. So we are not, of course, replicating that exact approach, but we are using the same mindset of finding a way to have our NK cells stimulating their own activation to be able to last for more than 2 weeks, up to 6 weeks or more. And we have screened on the right side, you can see actually only 23, but we screened 36 different activation domains that we integrate to our construct to be able to really enhance the activity of the cells and prolong that activity. And so far, we did that also at an ET ratio of 1 to 20 to put the maximum stress on our cells to be sure that when they are active, they are really active. And you can see a handful of those that are currently able to kill cancer cells. And I will encourage you to follow us at SITC. We have a poster presentation where we will say more, and not able to say that it's going to be presented somewhere else, but it will, and you will have the news coming soon. So in summary, I'm going to go through this very briefly. Why do we think we believe in our approach? The target is an incredible target and very appealing to be able to try to solve the problem of immunosuppression. The fact that the target is expressed in both tumor and tumor microenvironment and we can try to tackle this is key. The intracellular signaling that we have will enhance our cells is very important. And the fact that the target is expressed in multiple isoforms, that we are not facing that issue with our KIR construct is another add-on to our benefits. So allogenic is what we want to do, and that's the 5th point. But I'm running out of time, so I want to be able to conclude by showcasing that we are currently raising 15 million seed to be able to go to IND for AML and solid tumors. And we have some immediate need for cash also with a little bridge that we are doing currently. And we believe that with this, we'll be able to support patients. And as I always take a few seconds to always remember why we are here. Some of you may have heard the story. I have a friend, musician, that died of pancreatic cancer back in spring. And I just want to remind that we are doing this for the cancer patient. This is why we are here. This is why we wake up every day and do our work. I thank you for your attention, and I'll be willing to talk to you anytime.